not sharing. You guys see this screen? It should be right. Or is there like a big black box? May and Zoom do its job. There, all the boxes are gone. Anyways, today we're going to talk about which chapters are 8 and 9 and 10. Can't remember. But it's basically energy. The first, energy and the laws of thermodynamics. Wow. Okay, yeah. Catabolic pathways break down complex molecules into simple molecules and they release energy. Anabolic pathways synthesize complex molecules for simple energy and it consumes energy. You don't really know this. But you should know the concept. Anyways, forms of energy connect. Oh, who doesn't know this? Laws of energy transfer. Oh yeah. The first law of thermodynamics, second law of thermodynamics. I think this is pretty self-explanatory, so I'll skip that. And then free energy. Oh yeah, this is just exergonic and endergonic reactions. So make sure you know which one releases energy, which ones uses energy. If I'm going too fast, just tell me. Oh, energy coupling, fun. Some exergonic processes could power endergonic ones. Adenosine triphosphate (ATP) releases a lot of energy through hydrolysis of a phosphate into ADP. Ah, so that's how ATP works. Guys, remember that. Enzymes. Yeah, so enzymes they speed up reaction speeds. So they lower the activation energy, and that's what they do. And they love to test that. So remember that. And enzymes act on a substrate where it binds at an active site. There's an induced fit model, so it closes onto that substrate. So we have two types of competition. First, like inhibition. First, we have competitive, and then we have non-competitive. So competitive is you're literally competing with the active site. And that prevents obviously the enzyme from doing its job because the correct substrate is not in the active site. And non-competitive is when you bind to another site, which is also allosteric regulation. So you're binding to another site and you're changing the enzyme shape so it doesn't bind to the substrate. Also, high temperatures and unideal pH will denature enzymes. And there's also cofactors, coenzymes that will help the enzyme with electron transfers. Fabulous. Yeah, as I said, allosteric regulations. Yeah, and cooperativity. Substrate binds to a site of one unit subunit of an enzyme, and other subunits will be locked in active form. And you also have feedback inhibition. The metabolic pathway is halted due to product binding to the enzyme. First chapter is done. Chapter 9, cellular respiration. So over, yeah, you guys should probably notice. So first glycolysis, net gain 2 ATP to NADH. And here's the entire thing. You would probably want to memorize this. And yeah, and there's a reason why the Zoom password is G3P. It's because Derek and I both didn't memorize the glycolysis pathway and didn't get it correctly because it was G3P. And we only thought it existed in photosynthesis, which is wrong. So I should memorize G3P. The most tested would be glucose and then G3P and then maybe aldolase because that's what they produce with G3P. And then finally pyruvate. Don't really test anything else. Maybe these enzymes. But yeah, just remember like T3P exists in glycolysis, and remember the net energy, the net stuff. Like you get 2 ATP net, and actually you produce 4, stuff like that. So definitely memorize like where the NAD plus goes in, and where the NADH comes out, where the ATP goes in, ADP comes out, and then where the ADP goes in, ATP comes out. So you definitely remember those. Um, Next we have pyruvate oxidation. Pyruvate is turned into acetyl CoA, which represents coenzyme A. Pyruvate dehydrogenase. The diamine removes the CO2. Um, Lipid domain takes hydrogen electrons and transfers them to FADH2. FADH2 transfer to NADH, and product you get 2 acetyl CoA and 2 NADH. Fun things. The most important thing here is probably the coenzyme A. Coenzyme A literally goes in with pyruvate and forms acetyl CoA. Look who's here, Anthony. I didn't even have the link. That's crazy. Anyways, um, here's the thing again. It's found in chemicals. So, all right, here citric acid cycle. So the net gain also very important. 6 NADH, 2 FADH2, and 2 ATP. Like if you were to forget everything on this slide, remember one thing. Remember the net stuff. Okay. Um. So basically, 2 CO2 is generated per acetyl CoA. So there's one glucose that goes in, and it produces 2 pyruvate. So a lot of times they will say that, oh, there's a 6 carbon molecule and a 3 carbon molecule. You should probably know like which 6 carbon molecule it is and which 3 carbon molecule is, typically glucose and pyruvate. And then you know the pyruvate, one pyruvate forms one acetyl CoA. So one glucose molecule forms 2, etc. So you have to like understand kind of the thing that goes on there. Um, I don't. Yeah, here's the graph. I don't know why Angela put that there, but here's the graph. You should also probably remember all the. So I see. You should probably remember like acetyl CoA goes into citrate, isocitrate, alpha ketoglutarate, second CoA, secondate. Oh my god, man! I'm not gonna try to pronounce that. Man, Anthony bro shows up late and starts spouting nonsense in the chat. Anyways, malate, oxaloacetate. Like you should probably remember these two. Uh, Oxaloacetate, citrate, isocitrate. Like citrate, obviously that's why it's called citric acid cycle. So you probably should remember that. And then remember the like. I would assume people won't remember this. So the biggest takeaway is probably acetyl CoA goes in with, and then the final product would be oxaloacetate, and it forms citrate. You probably should know that. And then also know your net stuff, right? 6 NADH, 2 FADH2, 2 ATP, and then yeah, and then also remember it's only for one acetyl CoA. So for one, when one glucose goes in, is everything's doubled. Next, oxidative phosphorylation. This is where we find the electron transfer chain. So yeah, you wanted this thing. I don't know if Angela put it on here, but one thing you want to know here is ubiquitone, which is here. The Q. So everything else is us stuff you probably wouldn't know, but something that would definitely test is ubiquitone. Ubiquinone. Ubiquinone. I don't know. I always say in my head wrong, so I say out loud wrong. But anyways, the Q you probably should remember that. But everything else is cytochrome. So there are electron carriers between ubiquitone here. So you can see cytochrome B, cytochrome C1, cytochrome C, cytochrome A stuff. They carry the electrons to oxygen. Um, yeah, so here's the mechanism that happens. There's ATP synthase. You probably should know this from AP Bio or Honest Bio. Yeah, you see here like ubiquitone and you get cytochrome C. Cytochrome C is like really big thing in like evolution because everybody has it for some reason. So yeah, we'll talk more about that. But you can see hydrogen ions are passing through, and then they're going back to ATP synthase, generating ATP. And Andrew, I'm gonna mute Andrew, not Andrew, Anthony. Um, anyways, so yeah, I remember that. Wow, fermentation. Okay, so there's anaerobic respiration, which is different electron acceptor at the end of the electron transfer chain, commonly sulfur compounds, and you have fermentation. So anaerobic respiration would be like next to the high thermal vents, the little people over there. Anyways, and then your fermentation. You have alcohol fermentation and lactic acid fermentation. Fun fact: lactic acid is what makes your muscles sore after doing 20 minutes on the rowing machine. Uh, so in alcohol fermentation, that's also where you produce alcohol. That's why it's called alcohol fermentation. And then so if you have pyruvate, it goes in, and then you have glucose, and you go through glycolysis, producing the net 2 ATP, and then you have 2 pyruvate, and it goes in, and it turns into acetyl acetaldehyde, and then that's where they have the NAD plus regeneration into ethanol. So one glucose gets to 2 pyruvate, and then 2 acetaldehyde, and then 2 ethanol. And they also release CO2 in the process. So that's why when wine comes out, it's always bubbly, and they get rid of it. So um also producing ethanol is also really hard. Most of the time, it's also a ton of byproducts. So don't make your own alcohol at home, guys. Um, you will die. Anyways, and then also lactic acid fermentation. So you have pyruvate, and then there's no middle product here. It's just pyruvate, and then there's NAD plus regeneration, and it goes into lactate, and lactic acid. And then you have obligate anaerobes, which only use fermentation or anaerobic respiration, and they cannot survive in the present oxygen, while facultative anaerobes can do fermentation and respiration. All right, so connection of other pathways, as bullied here, beta oxidation is really important to know. Like, you cannot know anything else, but you have to know beta oxidation. So this is where fats undergo something, and it's broken down to carbon compounds for citric acid cycle. Here's the graph in Campbell's. Everything is from Campbell's. Campbell's is the Bible. I know you're not religious, but like, it's the Bible. Anyways, so you can see here proteins is broken down amino acids, and it can be inserted here to pyruvate or to acetyl-CoA or into the citric acid cycle, and it produces ammonia. And then carbohydrates go into sugars, obviously. And if fats either can be glycerol going to glyceraldehyde 3p. By its name, you can understand it's probably a subunit. And fatty acids, they go into acetyl-CoA and then into beta oxidation. Campbell 10, am I going too fast? I don't know. I'm only like 20 minutes in. I think I'm going a bit too fast. But we can do some practice. Anthony, do you have practice problems? So when I need Anthony to talk, he doesn't talk. That's great. No? Oh, well, you better go find some. Okay, so photosynthesis. So overview photosynthesis. Obviously, equation 6CO2 plus 12H2O plus light equals 6 C6H12O6 plus 6OH2O plus 6O2. But the equation won't be tested that much, but it's still, you should probably still know it, especially for people in AP Bio. Definitely know it. But there's two processes: the light reactions and then the dark reactions, which is also the Calvin cycle. And instead of NADH, they have NADPH and an NADP plus. Chloroplasts are the sites of photosynthesis, and stomata allows gas exchange, whatever. Yeah, there's like a stack of thylakoids, and they're stacked into a grana or granum, and they're surrounded by a stroma, which is the fluid. And then they're chloroplasts are typically found in the mesophyll, which is the tissue of the interior of the leaf. So first we're going to talk about the light reactions. So here, the lights or the photons excites electrons in chlorophyll or cardin. Okay, I'm gonna. I knew how to pronounce this, but I just forgot. It's a carotenoids. Man, Anthony's gonna be on my ass. But anyways, know this. I know how to spell it at least, so that's great. Carotenoids. There. Um. Okay, so you have carotenoids which use orange or yellow light, while chlorophyll is two types: chlorophyll A and chlorophyll B. Chlorophyll A is red and blue light, and the chlorophyll B they absorb violet or blue green light. Anyways, so the photosystems, the light reaction. So here, most important thing, linear electron flow. Okay, here's the notes Anthony gave me. Light harvesting center, carotenoids, accessory pigments, some chlorophylls catch photons and ferry energy to reaction center. Reaction pair of chlorophyll A molecules provide energies electrons to primary electron acceptor. Oh yeah. So basically remember it goes when the light enters the photosystem, it enters photosystem 2 first, and it excites electrons there, and it goes through to passed on into photosystem 1, and it gets excited again, and it passes on it, whatever. But in the next slide, you can see what happens actually. So directly quoted from Campbell. First step, a photon of light strikes one of the pigment molecules in the light harvesting complex of photosystem 2, boosting the electron to a higher energy level. An electron falls back to a ground state. An electron in a nearby pigment molecule is simultaneously raised to an excited state. The process continues with the energy being relayed to other pigment molecules until it reaches the P680 pair of chlorophyll A molecules in the PS2 reaction center complex. Excites the electron in this pair of chlorophylls to a higher energy state. Number 2, this electron is transferred to excited P60 and then the primary electron acceptor. Yeah, but basically what you want to know here. Why is Angela drawing? I'm gonna erase that right now. Whatever. Yeah. So light reactions. What you want to know: 2 before 1, and you also have cytochromes here. So they're passing down the energy, and they're being excited in these photosystems. And something you want to remember is P680 is in PS1, while P700 is in PS2. Everyone's so quiet today. Is it because Derek's not here? Um. Anyway, so next you have cyclic electron flow. So electrons are not passed into NADPH plus, but NADP plus, but are recycled through the cytochrome complex again. Hence, it being cyclic. So you can see here in the graph, they're being cycled. Okay, so next we're gonna talk about the Calvin cycle. So the Calvin cycle requires energy. So let's see. Calvin cycle. Oh, there's no graph here. That's crazy. Okay, anyways. So they build sugars from carbon dioxide or technically carbon fixation. So phase 1 is carbon fixation. So the CO2 molecule is attached to RUBP by the Rubisco enzyme. So really important stuff. In the second phase, you have reduction. So NADPH is reduced to CO2. No, NADPH is reduces CO2 into G3P. Remember that, please. That's why it's a password. And then every 3 molecules of CO2 is turned into 6 molecules of G3P, but only one G3P exits the cycle to be made into sugar. And then since G3P, since only one one GDP gets produced, you can see like later on you need to cycle through many times to actually go produce some type of sugar. Um. Anyways, so stuff you really need to remember here is definitely 3 CO2 goes in, it gets turned into 6 C3P, and this 6 C3P one leaves, and the other 5 produces 3 RUBP, and it gets attached to 3 CO2 again. So this stuff you should remember. Yeah. I don't know what this photo is for. I guess that's for, oh, that's not Calvin cycle. It should have been before the slide. I might have moved it by accident. Here, alternative methods of carbon fixation. Stomata trade-off. You have open stomata. The water lot is lost by transpiration, but you also can get carbon dioxide for carbon fixation. But when you close stomata, you conserve water, but you also don't get enough carbon dioxide, and you also can't get the oxygen out, which is often poisonous for plants because they produce oxygen. And our bisco binds to oxygen instead of carbon dioxide, and it wastes energy because they produce a side product that is useless. So what you see in there's adaptions, except for example, will be C4 plants where you have kind of like a bundle sheet cell. You have bundle sheet cells, so you have two different cells that contain each thing. So you have one cell, that's the mesophyll cell. Carbon dioxide gets converted into PP carboxylase, and that gets turned into, follows kind of the cycle, and a malate, which is 4-carbon compound, gets converted into pyruvate, a 3-carbon compound, and carbon dioxide in the bundle sheet cell, and the carbon dioxide goes through a Calvin cycle. So there's like an extra zone for, because C4 plants live in environments that are often harsh and hot, not enough water, etc. So they need this extra layer of, protection and complexity in order to save water, but also generate energy during the day. Um, so and also can plants, they take in CO2 during the night, and they store CO2 in the night as organic acids, and they released it in the morning. So they can continue going through the Calvin cycle during the morning when their stomata is closed. Oh, so here is the graph I was looking for. So yeah, you can see here, CO2 goes in and gets using Rubisco enzyme. It gets converted into some short-lived intermediates, and it gets converted to GDP in reduction. And then only one GDP is the output, and then everything is cycled back into RUBP, which is then added to the 3CO2. So 3 RUP plus 3 CO2, and then the cycle continues. So kind of remember, like a lot of the tests and stuff, they love like playing with these numbers. So they'd be like, oh, how many would you get with, this many carbon dioxide molecules or this many G3P molecules? So you gotta remember the numbers, especially for the carbon dioxide, G3P, and you gotta remember like how many carbons are in these things. So carbon dioxide, obviously 1 carbon, but G3P you have 3, etc. At the end, wow, okay. And the other slides, the other problems.